Good afternoon, everybody. Welcome to our, I guess, the first grand round of the new year. So happy New Year to everybody, and we're kicking it out. We got burgers in the back there. So if you haven't eaten and you're here in person, go get a hamburger. And where's our Smash Burger? I think is where we were from. So Smash Burger back there. Today I have the pleasure of introducing Dr. Ezra Cohen. I've known Ezra for a long time. I don't know how we met. I have no idea. But from through ASCO.
On a panel or something.
From ASCO panel,
yeah,
I think. But and Ezra is Chief Medical Officer of Oncology at Tempus. He had a distinguished research career before going to Tempus. Most recently, he was Chief of Division of HEMONC and Associate Director of Clinical Science at UC San Diego, and still is in San Diego. Dr. Cohen also led the Precision Immunotherapy Clinic and co-directed the San Diego Center for Precision Immunotherapy, UCSD. Prior to that, which I think I met him when he was at University of Chicago, he was a co-director of the Head and Neck Cancer Program, as well as Hematology Oncology Fellowship Program Director. Again, welcome, Dr. Cohen. He's going to talk to us about partnering to power precision in medicine. Thank you so much for joining us.
Thank you, Craig, and thanks for the invitation. Thanks to you folks for coming. Can you hear me in the back?
Yes.
Yeah. Okay. Great. So, as Craig said, we're going to spend probably about the next 45 minutes talking about what we've developed at Tempus, and hopefully, if you don't know us well, hopefully enlighten you guys on what we're trying to accomplish. And I think you'll agree that it is part of the future of healthcare. If you do know us well, this may be redundant, but hopefully you enjoy it. And I should say, I'm going to tag team the presentation a little bit. I'll bring up Eddie to present a couple of slides, and then Renee and Joe to talk to you about some of the AI applications towards the end. But let me start by sort of giving you the overview of the reason Tempus exists and the vision from the beginning. We started by doing diagnostics, and many people still view Tempus as a diagnostic company, and certainly and I'll show you, what we're doing in that area. But right from the beginning, the idea was that if you could do the diagnostics and pair that with clinical data, so you have molecular data that's clinically annotated, you could build a database that would be incredibly useful to researchers, to pharma, and really begin to move the field forward, and also would help with decision making from the provider perspective. About 5 years ago, and we were anticipating this, although we didn't know when it would happen, the large language models became really good. And what the large language models in what now is looking like the third wave of AI, what they're able to do now is take multimodal data, and you see that in the black circles at the top, integrate it, irrespective of the format, doesn't matter what type of file it's in, but make sense of it, interpret it so that we can put it into the database. So the diagnostics led to the data. The data has allowed us to build AI applications. And I'm going to give you examples of all that through the talk. We're not a small company anymore. We now, and we're operating, certainly at scale. About 65% of academic medical systems in the US are now connected to Tempus, including you guys. You're about to go live any day, I'm told, hopefully next week. About 50% of oncologists will regularly order with us. Believe it or not, we now have 40 million records across all products and services. These are individual patient records. I'll talk to you about the database in more detail in just a second. And we have now almost 4,000 employees. Last year, we ran 1 million next generation sequencing tests. So we're, again, operating, certainly at scale. And here's really the idea that I'll reiterate again, is that what we set out to do, and you'll excuse me, I'll cough from time to time. I picked up RSV about a month ago. I cannot shake this cough, so my apologies for having to interrupt, but don't worry, I'm not contagious. What we really set out to do was transform care delivery. And we thought that technology through AI, through the large language models, was going to be able to do that. That's through delivering high quality care with precision. We'll talk about that in terms of the testing and some of the other applications. Harnessing innovative technology, and we'll talk about that certainly a lot more, and then leveraging data, which, by the way, we'll present to you, or Eddie will present a couple of projects that you guys have done using our database, very, very interesting presentations. And I'm going to divide the talk into the diagnostics, the enabled care, and the enabled research. So with that in mind, this is basically the diagnostic menu. This is a very busy slide, but let me spend a couple of minutes just to sort of show you what we've tried to build. And the idea behind it was that we wanted to offer providers one place to go to essentially get anything that they need along the cancer patient journey. That has obvious advantages to the provider, to the patient, in that you don't have to get results from different labs, and it has obvious advantages in terms of being able to build the database. And so at the top left is the workhorse of the whole system, and those are the XT tests. That's the next generation sequencing, 648 gene panel that really has all the relevant cancer genes that one would want. Several years ago, I think about 5 or 6 years ago, we introduced whole transcriptome, and now we've been running whole transcriptome regularly. I'll show you where that comes into play. Of course, we have liquid assays as well, the XF and the XF plus being 523 genes and able to have blood TMB on that as well. We do have, we acquired a company called Ambry about a year ago. Ambry now is our germline offering. So we do offer germline susceptibility testing for oncology. You can see that on the other side of the slide, Ambry also offers tests in rare disease, not necessarily applicable to this audience, but more than happy to answer any questions around that if you're interested. Recently, we introduced a pharmacogenomic panel by acquiring a company called OneOme. So now we offer DPYD, UGT1A1, and other relevant pharmacogenomic variants that would apply to cancer therapies, and one can order that with about a 4-day turnaround. You can see, I'll talk about the AI-driven algorithms just in a few minutes, but we also have now begun to offer immunochemistry tests that are relevant to mostly to antibody drug conjugates. So again, the idea is to be able to offer testing parts of that patient journey, including, by the way, MRD that we'll talk about in just a few minutes. Excuse me. So we wanted to take that. So that's the diagnostics, but the diagnostics themselves were not going to be enough to really give the optimal product to providers. And we wanted to couple that with smarter insights. And what we've been able to do, as a few examples here, is because of the comprehensiveness of the panel, we've been able to notice that 30% of patients will have alterations associated with an FDA-approved drug that are only detected in RNA, underscoring the importance of the whole transcriptome. As you can guess, the majority, actually all of these, are fusion events. We've been able to notice that a reduction in false positive rates when we match the normal sample to the XT, the solid tumor. We were the first to ask for a normal sample because we felt that it would improve the accuracy of our testing. And in fact, it does. It reduces that false positive rate by about 30%. But the other thing it did when asking for the normal is that it uncovered unknown germline pathogenic alterations that would have made that patient susceptible to cancer. And when I say unknown, these are patients who did not have a typical personal history or family history, and they came in cancers that you wouldn't normally think about with respect to germline alterations, things like cholangiocarcinoma, head and neck cancer, esophageal cancer. And it turns out that about 7% of patients have these. So that's a little bit more than 1 in 20, and we think that that's significant. And then we've been able to show that the idea of doing both solid and liquid testing simultaneously also has merit. Here, about 10% of patients will have an alteration found in liquid only that is linked to an FDA-approved therapy. And that's 1 in 10 patients who would otherwise not get the therapy if that test wasn't ordered. We introduced last year something called comprehensive therapy selection across multiple tumor types to make ordering easier. This is a panel of tests that would normally be ordered for that specific cancer. So instead of having to check each test individually, one can just go to their cancer of interest, or cancer that's relevant to that patient, and check that CTX box. And then right from the beginning, literally from day one, the philosophy was that we were not going to make financial restraints a barrier to patients getting these tests. And so we introduced a financial assistance program. This is the form, it's literally one form, where a patient, if they are approved, and I can tell you that the great majority of patients get approved, over 95%, they either have no cost over an 18-month period or a maximum of $100. We have to put that in place by law, but what we've tried to do is make it as simple as possible for at least eliminate any financial concerns as much as possible for patients to get these tests. And the provider can order as many tests as they want over that 18-month period. And that 18 months can be renewed ad infinitum. So literally, if you wanted to order a blood test, a ctDNA on a weekly basis, I don't know why you would do that. I'm not suggesting you do that. But if you did, it would be covered by the financial assistance program. So with that, and by the way, feel free to stop me along the way if there are any questions. Happy to stop talking and answer questions. But let me talk to you about now, I alluded to the algorithms. And what I want to show you is a couple of examples of the algorithms that we built. Again, the diagnostics got us the data allowed us to build the algorithms. And what we set out to do was build algos that would have clinical utility. What you see in the middle of the slide is, and we've just recently introduced Page Predict. I'll talk to you a little bit about that. A few months ago, we acquired a company called Page AI, a digital pathology company, and we've been able to incorporate some of their predictive models into our testing. Ironically, our first AI approval came in cardiology, and although I won't talk about this in any detail, but more than happy to answer any questions, we've got a couple of electrocardiogram AIs, one that predicts atrial fibrillation, and the other one that can demonstrate low ejection fraction. Again, those are FDA approved, and more than happy to discuss them, but I'm not going to go into detail. So really AI, we are an AI company. So that's, if you leave here with one thing, just remember that, that we're not a diagnostic company. We have diagnostics, but really we're an AI company. And we deliver AI by those smarter insights that I talked to you about, the digital path insights that I'll talk to you about a little bit, the algorithms, I'm going to show you a couple of examples, and then the connectivity that you'll hear about towards the end of the talk. So let me talk to you about one of the algorithms that we developed. We call this IPS, or the Immune Profile Score. And basically what we did was we set out with a simple question. Could we use our database to create a model that would be predictive of immunotherapy efficacy across solid tumors, so pan-tumor, in either first-line or second-line therapy, either as monotherapy or in combination? And obviously the answer is yes. or I wouldn't be presenting this. And that's what IPS, or the immune profile score, became. This is what the result looks like. It's actually a number. There are no units associated with the number. It goes from 0 to 100. It is actually a continuous variable. So the higher the score, the more likely that person is going to benefit. But for ease of use, we dichotomized it into either IPS high or IPS low. Across 19 solid tumors, it turns out that about 2/3 of patients end up being IPS low, and about 1/3 of patients end up being IPS high. Does it matter? We think it does. And here are some of the data. We published this a little bit more than a year ago in JITC. Happy to share that publication if people are interested. But not only does it matter, not only does IPS predict survival in patients getting immunotherapy, again, first line, second line, monotherapy, or combination, it does better than existing biomarkers. And here's what I mean by that. If you look at the TMB graph, what you can see is that we can identify patients who are TMB low, that's the aqua or light blue, but IPS high, that do relatively well with immunotherapy. By the same token, we can identify patients who are TMB high, so approved for immunotherapy, across the board approval if you're TMB high, but if you're IPS low, you don't do very well with immunotherapy. If you go to the middle graph, so that's microsatellite instability, interestingly here, if you're MSI high, your IPS score doesn't actually matter. So IPS low, MSI high does just as well as IPS high, MSI high. But if you're MSS stable and you're IPS high, you actually do really well with immunotherapy, almost as well as MSI high. Again, that's the light blue or the aqua line. And what I'm not going to show you in this deck is that we just submitted to ASCO an abstract looking in colorectal cancer, microsatellite stable colorectal cancer. And we had the exact, actually even a little bit better hazard ratio between IPS high and IPS low, a hazard ratio of about 0.3. We'll present those data at ASCO. So even in microsatellite stable colorectal cancer, IPS can differentiate patients who will do well with immunotherapy. And then lastly, PD-L1, sort of the gold standard biomarker that I think we'd all agree is good, but it's not great. What you can see from these graphs is that IPS can find a group of patients who are PD-L1 negative. Again, that's the light blue. PD-L1 negative, who do very well with immunotherapy. And the converse is also true. PD-L1 positive patients who do poorly with immunotherapy. So we really think that there's value to the score. And we've got good uptake. John.
Is the IPS on your tissue assay, blood assay, both?
Oh, thank you for asking that. It is a tissue assay exclusively. So it was built on both DNA and RNA. So you'd have to order both the XT and the XR, and then check the box for IPS. Yeah, it's not a blood-based assay. Yeah, I should have mentioned that, and thank you for that question. So yeah, please.
The IPS, like what constitutes the IPS?
I can't hear you, Sean.
What constitutes the IPS?
Yeah. Okay. Thanks. What constitutes it? For the sake of time, the question was what constitutes the IPS. For the sake of time, I didn't put that slide in. Again, more than happy to share the publication. But essentially, we started with multiple features that we tested in the original model. And then we came down to one genomic feature, TMB. That's the only one that stood out. And we could not take TMB out of the model. If we took TMB out of the model, it was no longer predictive. But mostly it's an RNA, so transcriptomic signature. We came out with 3 sort of global RNA signatures that have been published. One around myeloid cells, one around T cells, and the other around, I can't remember what the other one is, but again, I'm happy to share the publication. And then the rest of the RNA was individual genes, including PD-L1, so CD274. So those are the genes and signatures that held in the model, and then in the validation set, they continue to have predictive utility. Did I answer your question? Great. So moving from immunotherapy to HRD or homologous recombination deficiency, we now recognize that there are therapies for patients whose tumors are HR deficient, and there's some value in demonstrating those patients. And so we set out to build a model that could find these patients even if they were BRCA negative. And again, we were able to do that. We call either positive or not detected. And here you see that the model is working in both ovarian and breast cancer. This was the first iteration of it. And then what we were able to do, and this was purely a DNA-based model, what we were able to do, oh, and I'm not going to show you that. So sorry. What we were able to do recently, and we're just publishing this now, is develop the second version of the HRD that is now RNA-based and has retained the predictive value in breast and ovarian, but has now improved the accuracy in prostate and pancreatic cancer. And again, we're presenting that abstract, the pancreatic cancer part, we're presenting that abstract at ASCO. Hopefully it gets submitted. So now we have the ability to call HRD across 4 diseases where this commonly happens: ovarian, breast, prostate, and pancreatic. Again, based on the internal database that we've been able to accumulate over a decade. And the last thing in terms of AI that I wanted to talk to you about is digital pathology. So many of us, and including I'm sure you guys are interested in DigPath. We think it's part of the future of diagnostics and decision-making in oncology. In fact, we thought so strongly that we acquired Page AI again a few months ago. And the strategy really here is integrate digital pathology as another modality in what we have that includes DNA, RNA, and blood testing. And here's what we call the third revolution, if you will, in pathology. So it started, of course, with the microscope. In the early 21st century, digitization began, and now computational pathology that can pick up features on an H&E slide that are not available to the naked eye. Integrating digital pathology requires, obviously, tissue stewardship and digital biomarkers. But the way that we've been able to incorporate it, before I get that, the way we've been able to incorporate it is with something that we call Page Predict. And that we literally just launched 2 days ago, where what we're able to do is predict from an H&E slide the chances of that patient's tumor having a specific list of alterations. And the way we've incorporated this is that we know that in a certain number of, especially non-small cell lung cancer and prostate cancer, because of the amount of tissue that we receive for these samples and the type of tissue, there is a fairly high QNS rate, or quantity not sufficient. In fact, in non-small cell lung cancer, it can be as high as 10%. And we thought, rather than asking for another tissue sample, which of course we could, but sometimes in patients it's difficult to get, could we use digital pathology to at least give the probability of specific alterations that might be targetable? And the answer is yes. That's Page Predict. And what Page Predict can do is, again, based on digital pathology analysis of that H&E slide, it will give a listing of alterations and their probability that they occur in this person's cancer. So based on that number, a clinician can decide, is it worthwhile trying to get another biopsy? Is it worthwhile sending a ctDNA test? Or if that accuracy is so high, do I make a treatment decision based on it? So at least we can give some sort of answer when a provider sends us a specimen, even if it can't be the sequencing itself. So let me move from there to the research and collaboration component. And what I'll say, a couple of things to this audience is first, we are very interested in collaboration, and we are very interested in working with external investigators. You guys are actually a Tempus Plus site, which means that you have access to our database that we call LENS, and you have preferred access to the Discover program, which is our gateway for external investigators. So I would encourage you to use it. What this slide shows is that we're more than happy to provide the raw data to you if that's of interest. If you want, we're also able to do our own analytics and bioinformatics in-house. If you have an idea that requires that and you're, for whatever reason, unable to do it, we have personnel that can do that as part of the Discover program. So this takes a few different avenues. The first is you bring ideas to us. That's Tempus Discover. That's probably the most common way that external investigators work with us. It's literally a one-page form. You'd work with your medical science liaison to basically fill out that form, generate the hypothesis, the aims, et cetera, et cetera. That form gets reviewed on a weekly basis. And I can tell you that the great majority of them get accepted. The biggest problem is if it's not feasible, if it's something that we just don't have the data, we can't do. But other than that, we try to accept as many as possible. These can take the form of research grants. We're more than happy to collaborate on grants that you're submitting. It can take the form of independent research, and basically it's a one-form submission. The other way is we bring ideas to you. And now as a Tempus Plus site, you'll be getting more of these, I hope. And that we call the Sparks program. So the Sparks program essentially means that we have an idea that was generated internally. And we looked at our data initially to see if the idea had merit. And indeed, it looked interesting. And now we need external investigators to help us further that hypothesis. And that's basically Sparks. We would come to you, we'd say, hey, are you interested in doing this? Is this within your area of interest? And if the answer is yes, we will work with you on it. And then we work on ideas together. Those are study collaborations. Example of these are MRD, algos, and digital pathology. And this is really very much collaborative, really almost doesn't matter where the idea is generated. The point is that we do it together. Here's an example of a Tempus insight. So enrichment of copy number loss events among patients with breast cancer and ESR1 gain of function mutations. So you can see this is a Spark. We looked at our database for ESR1 gain of function mutations. And indeed, we see different, you see the mutations listed in the table there or the graphic. And what you can see is that, in fact, we do see these gain of function mutations, coincidence with loss of wild type ESR1. But this is just the Spark. And really, I think to make this interesting, we need external collaborators to take this even further. Here's another example. This is androgen receptor and PSMA co-expression in metastatic castrate-resistant prostate cancer. And you can see that there's a difference in those expression levels, especially with respect to different genes such as DLL3. And again, an observation that we were able to make in our database that we'd love to take forward with external investigators. Here's a third one, circulating tumor DNA, EGFR positivity associated with poor outcomes to osimertinib in non-small cell lung cancer. And so what we were able to demonstrate here is that the median survival on patients on this EGFR inhibitor actually was lower depending on the quantitative level of ctDNA of the mutated EGFR. Again, an observation, we think it's interesting. There may be investigators that want to dive further into this. So we provide access, and now as a Tempus Plus site, you have access to the multimodal Tempus database that we call LENS. It's useful for hypothesis generation, research collaboration, and joint publications. There are there are now about 9 million individual oncology patient records in that database, 9 million. But the real golden nugget of the lens is about 500,000 patients, individual patients with DNA, RNA, digitalized images, and longitudinal clinical data. And that longitudinal clinical data includes things that you would expect, the patient characteristics, the tumor characteristics, the stage, things like that. But it also includes therapy. It includes the sequencing of therapy, how long patients were on specific therapies. It includes overall survival. It includes comorbidities and things that really can enrich a hypothesis and a dive into the database. So you guys have access to it. I'd encourage you to use it. So I won't belabor this. I think I made the point. So Eddie, I think you're going to present a couple of examples that came out of MUSC now, and I'll let you.
Thank you. Yeah, thanks, Dr. Cohen, and thanks for letting us present today. I thought it would be amiss if we didn't highlight something really important that came from the San Antonio Breast Conference Symposium. We were thrilled and excited to work with Abby Siva on 2 posters that were accepted. As Dr. Cohen noted, our database allows for providers to come to us with specific questions and look at cohorts of interest. So Dr. Siva came to myself, and we have a specific external research MSL as well, to look at a cohort of patients that needs consideration. So patients, metastatic breast patients that are hormone receptor positive, HER2 negative, that are co-mutated PIK3CA and ESR1. This happens in about 15% of cases of pretreated metastatic breast patients. And we're still trying to work out some of the information about the sequencing of the therapy. But Dr. Siva came to us and asked, could we look at this cohort and come up with some insights from that specific cohort in our database? And we were lucky enough to have over 8,600 patients that were the metastatic breast patients, hormone receptor positive, HER2 negative. And we had a sizable proportion as well that were co-mutated, over 500 patients, co-mutated ESR1 and PIK3CA. So this poster specifically focused on the DNA components and then looking at real-world overall survival as well. You can see that if we look at comparison of wild-type patients versus PIK3CA mutated only versus ESR1 mutated only versus co-mutated, there certainly were some different characteristics. We also saw that there was a higher proportion of TP53 mutations as well. So maybe there's kind of a triple positive, TP53, ESR1, PIK3CA. And we also noted that the real-world overall survival was certainly shorter for that co-mutated population as well. I think it just adds that we can bring some of these insights and learn a little bit more about that. And the reason that we can do that is because we have such a larger database that has both DNA, RNA, and the clinical data as well. So a really nice poster. We were able to use both our tissue samples and our liquid biopsy samples in this poster as well. And I'll just go to the next one. Sorry, the next one focused more on the RNA component and sequencing piece. We were able to look at differential gene expressions for this cohort of patient and also the tumor immune infiltrates as well. And I think the goal here was to think about some therapy implications down the road. Are there differences, especially in that immune infiltrates, that may be different for that co-mutated population versus wild type versus some of the single variant patients as well? And I think what we saw is that there were some distinct transcriptomics and immune phenotype signatures. We did see that the co-mutated group and the PIK3CA mutant group really had a higher level of those immune infiltrates. Maybe they warrant more consideration in the immunotherapy realm, whereas the ESR1 mutant only population did not, maybe more so from different type of endocrine therapies. So a really nice example. We always want to show what good looks like. We're really happy to partner with Dr. Siva. I'll note just as I finish that we were standing across from another group at Henry Ford. They're another Tempus Plus institution. And Dr. Siva, Dr. Baldick, Dr. Daybach from those two groups have now connected to talk about how do we move forward. They had a small cohort that did look at some of the timing of the sequencing of the therapies. So it's really nice to see folks collaborate together. So thank you for letting me present these posters. And we were, again, thrilled to work with Dr. Siva.
Great. Thanks, Eddie. And it really just underscores what one could do if you had an idea. And even for a relatively uncommon co-mutation event, we had 500 patients in the database. So you can begin to make something meaningful out of the data. And of course, this has therapeutic implications, which we're always excited about. So again, I think we've been over this. I'm not going to belabor it. Essentially, the research collaborations take primarily two forms. If you have an idea, that you want to explore with us, we'd love to hear it. We'd love to work with you. We're generating ideas, and we'd love to bring those to you if you're interested. So with that in mind, really, I'll finish with this slide. I'm going to turn it over to Renee and Joe, or both or one. Both, okay. To talk about a little bit more about the AI and connectivity. What I really wanted to illustrate is that we're trying to be as comprehensive as possible. We're trying to deliver those smarter insights, develop the science, develop the technology, because we feel that this is the future of healthcare and certainly the future of oncology. Our whole reason for existence is to reduce uncertainty with information. You can imagine, and because you're in the clinic, most of you are, some of you are, that every time we see a patient, there's always going to be some uncertainty with that decision. What we're really trying to do is reduce that uncertainty so that providers can make the best decisions and life science companies can make the best decisions, which is something we didn't talk about too much, but they can make the best decisions when developing drugs. So with that in mind, let me move to the last part of the talk, and that's Tempus AI applications, and turn it over to Renee.
Yeah.
Thanks, guys.
So as Ezra was talking about, we have all of that really great stuff on the clinical side that we are leaning into and working with. We also have a whole other side of Tempus that focuses on AI applications and being able to take a lot of the data that we've worked over the years to accumulate to be able to provide insights back to researchers and physicians to take care of patients. And so we're going to go really high level over this, probably about 5 to 10 minutes, because we do have a meeting later today to go really in depth into this. We could probably spend an hour and a half talking about all the different things we have on the AI application side. So we're going to go a little bit more in depth around 3:30 later for this meeting. So we're going to go high level over what we're scoping out with MUSC and what's currently in flight with you guys to be able to provide some more support for your patients. And so
One say, I just wanted to add in here, just to be clear too, a little bit different from the Tempus Plus program, which is definitely more research centric. These applications are more patient centric in the sense of it is true PHI, it's true insights to impact care and research from a identifiable data perspective. So it is a little bit different from our Tempus Plus and Lens applications. So just wanted to kind of make that clear that this is more, again, clinical decision support and medical device type of tooling and applications.
So on the left, you can see the clinical intelligence. And so what Tempus has really leaned into is being able to create different tools that support clinicians with care pathways for patients to be able to deliver guideline-based care to assist patients to not fall into some of those care gaps that we often see, particularly in oncology. But as you can see, we've also been able to assist with areas like cardiology and IPN, so the incidental pulmonary nodule. So we have different abilities to create pathways to support physicians and clinicians with patients in that area. And then one of the areas that Tempus has leaned heavily into as well is clinical trial support. So we do know that a lot of academics, and particularly MUSC, has a large focus on clinical trials. And so we have 2 different arms that support the clinical trial initiatives. Our services arm, which includes our time program that we are currently working on with MUSC, and we'll talk about in a minute, a little bit more in depth, and then our technologies. And so our time program is our white glove matching program that allows rapid activation and helps find eligible patients for clinical trials that we can then bring to your site in a rapid fashion and open quickly to be able to provide those novel therapies to patients. We then have our clinical trials technology, which includes a lot of our different AI-enabled platforms that essentially help with self-service cohort building tools to help match patients for other clinical trials, not in that time network, but other trials and initiatives that you have at your site as well outside of the time network.
Any questions on that?
So time is, so it's one of the largest, if not the largest, data integrated clinical trial network in the nation. So we work with sponsors to bring a portfolio of clinical trials in to provide to sites to rapidly activate for patients that are eligible for those trials. So we've essentially flipped the script of what you guys are used to with traditional clinical trials of working to activate a clinical trial that takes several months, go through all the negotiation process and everything to get a clinical trial up and running, and then look for patients that match for those trials. We have the ability to help with identification of patients prior, and then move quickly to activate that trial for those eligible patients. And so we have a lot of different operational parts of our program that are standardized that allow us to do that with the sponsors that bring their trials into our network, and with sites that want to participate in the time program. And so, as I said, we're currently working on this with MUSC. We're working to be able to bring that portfolio of clinical trials to your patients so that we can get those trials up and running quickly, and then we can support the matching services that go into finding patients for those trials and being able to get them enrolled. Anybody have any questions about that? So we have, as I said, the time program is a rapid activation program. So we have two different models that we support for clinical trial activation. So our just-in-time model is our very fast model where we move to open a clinical trial for a patient that is eligible. With community sites, we typically do this in about 10 to 14 days. So we're able to identify a patient and get that clinical trial up and running in about 10 to 14 days, and then get a patient consented very shortly thereafter. We typically, with academic institutions, specifically NCI designated cancer centers, we know that there's some things that go into that process that make it a little bit longer. So we're typically around maybe 4 to 6 weeks to get those clinical trials up and running, but it's still incredibly fast when you're talking about trying to get a trial open for a patient that's in need. We do support the more traditional clinical trial model that you see at the top of the screen, the expedited. You'll hear us call it expedited or prospective, and that is where we're activating a trial in lieu of patients coming onto that trial. We just do that very fast as well, but we also do that based on the data that we're able to help with finding patients. So we don't want to open clinical trials at your site that are not relevant for your patient population. So we're able to help find trials that are relevant for your patients by looking at the data and saying, you have this population, this is a great trial that would match for your patient population. We can work to get this trial up and running. Does anybody have any questions about that? Great. So that.
I was going to add one last thing. So everything we kind of showed you so far is a lot of our applications and toolings and technologies that we can provide more out of the box or directly into the clinical workflows. I think one thing just to also consider with Tempus is we're really at our core a multimodal data platform. And why that's critical is a lot of oncology data, specifically precision oncology data, 80% of that data is usually unstructured. So we spent the last 10 years kind of developing a platform that harnesses a lot of that data. So whether it's in a progress note, in a pathology report, if it's scanned into your media tab on Epic, we're able to acquire and utilize that data for those different types of insights. So whether it's supporting applications that we can provide, or if it's supporting other initiatives that you have in your institution, if you need just better and clean data, this is an area that we have expertise in that we can help support other projects and initiatives in addition to the applications we can serve immediately. So just food for thought, again, if there's other considerations or use cases that you may have where you need that kind of rich, clean data, especially in precision oncology, that's an area that we can help support. Any other questions for us?
much is like, if I order tests on a patient's tumor, how much is this sort of a living document thing? Because as you know, you guys are developing new things and all of a sudden it's like, okay, that wasn't there. That wasn't there when I said, that was not part of the technology offered when I ordered this a year ago. And there can be some liability things associated with this thing. Oh my gosh, Dr. Blackrock, you didn't see that they had a, Dr. Cohen's famous immunotherapy thing that at the time. In fact, you can, those kind of stuff. How much, and how do you do something along those lines in, a company like yours where the result that you gave me 2 years ago, that it's already in there. How does that work in something like what you guys are doing? I don't know if I'm articulating it well enough, but
No,
as you know, that happens.
So we address that with something we're just about to launch, like literally in the next week or two, called Tempus Refresh. Do you mind if I answer?
Yeah,
Please.
Yeah, this is your domain for the media. Please.
So
I was thinking the same thing.
to Craig, exactly address what you just said. So a great example is you may have a patient with non-small cell lung cancer that has a HER2 tyrosine kinase mutation. A year ago, there was no drug approved for non-small cell lung cancer with that alteration. Now there is. What Refresh does is essentially it refreshes the report so that it notifies you that, hey, this is a result that you had, whatever it was, a year ago, 2 years ago. There's now an FDA-approved drug for this alteration. More than that, if there's a VUS that becomes known, a known significance, so it goes from a VUS to a targetable alteration, Refresh can do that. Version 2 of Refresh, and this will be probably 6 months from now, will also surface clinical trials that are newly opened, new for that alteration or for that patient, that again will notify the provider, hey, when you ordered this test, this clinical trial didn't exist. It now does. You might want to consider it. The other thing that we've done, aside from Refresh, is so for instance, for IPS and for HRD, the algos that we developed along the way, we've gone back and we've asked providers, hey, this algo is available. We're happy to run it on your patient. We have the data. If you want the result, just let us know. We did that actually for, believe it or not, 80,000 patients in the last 6 months. So there's ways that we try to keep up. It's obviously relevant.
Quick question. Oops, sorry.
Dr. Siva, yes, go ahead.
Okay, thank you. Sorry, virtual question. So thank you for the great presentation. And I had a quick question about the predictive markers that you're coming up with, the immune prediction as well as the HRD markers. These are all developed from retrospective data, but the question is, like, currently in the triple negative setting, very small proportion of patients are getting immunotherapy or benefiting from PARP inhibitors. But if you can identify more patients who could benefit, I think that would be definitely helpful. But do you think a prospective validation is necessary to get that far, especially to get insurance to approve these drugs in that setting?
Did you hear the question? Sorry, I had a hard time hearing. It was MRD related.
Yeah. Can you ask that question again? I'm sorry, it's a little bit hard to hear.
Okay, so this is regarding your immune prediction model and the HRD model you discussed. Can you hear me now?
Yeah.
Yeah.
The HRD model.
Yeah, the question is, are you planning to do prospective studies to validate this so we can get insurance approval for these markers? Because currently, I don't think like showing this marker alone would let us use these drugs in those patients.
Yes, 100% agree. And the answer is yes, we're actually working. And if you're interested in being one of those investigators, we'd be happy to talk to you further. We're talking to investigators right now about HRD. We're also talking to investigators about validating prospectively IPS. So yes, we realize the same thing, that we will never get, we'll never be able to demonstrate full clinical utility without some prospective testing.
Yeah, and I would be interested. Yeah, we can talk further.
Great, wonderful. We'll follow up.
Alan?
Just curious, like for the data that you showed from the breast conference and clinical correlates, like where do you get that clinical data from?
Where do we get our clinical data?
Yeah, is it just the like notes and pathology submitted when someone puts it on?
So for the 8 million plus or the 9 million patients, the majority of those are when a provider orders a test, they submit a pathology report and a progress note. Excuse me. So that's the majority. The 500,000 with the longitudinal clinical data, those are EHRs that we are connected to, that we exchange data with literally on a daily basis. I don't know, Joe. If you
Yeah,
want to-
that's correct. So it's split. So some sites are connected with us just for ordering and resulting. And to Ezra's point, we do pull the most recent progress note that gives us the histology. And it also gives us, and we also pull the most recent pathology report. So it gives like a snapshot in time for that patient. But then we do have a significant amount of sites that are using a lot of our AI applications where we have a more longitudinal data record connection to their Epic system. So we are pulling the full comprehensive medical record.
Giorgio?
The digital images, do you use resonance for as a model for determining the tissues, like to validate your tissue diagnosis? Like which model do you use for digital imaging?
What models are we using to build the digital path AI? I actually don't know.
I can get back to you on that. There's been a lot of changes with our acquisition of Page. And I think we're adopting new models versus maybe the models we've already have existing. So I can get back to you on where we're at with that. Thank you.
There is Dr. Jane online who has a hand up for a question. Dr. Jane.
Yes, good afternoon. Thank you, Alicia. Thank you, Dr. Cohen, for the overview of Tempus platform. I have a couple of questions. One is, let us say I'm a pathologist, standard routine pathologist, looking at an H&E of a breast cancer sample, ER/PR positive, HER2 negative. Can this platform make a diagnosis of that ER/PR positive, HER2 negative breast cancer, and perhaps even identify HER2 low or ultra low status? In other words, could the need for H&E be replaced with simply a diagnostic molecular platform that you offer? And then I have another question.
So if I heard the question correctly, can the DigPath predict HER2 status? Is that essentially the question?
No, not predict, diagnose, identify. In other words, could you take a tissue sample and make a diagnosis of a specific entity using exclusively the molecular platform, therefore eliminating the need for H&E staining and the need of a pathologist, for example?
I can't hear the question.
So basically, if I just send you a piece of tissue, can you tell me what it is? No, I don't give you any anatomical information or nothing. If I give you a piece of tissue, can you tell me what that is and everything that I would need to know about making, I mean, I get the amount of data that you have generated that you should be getting that more.
Yes.
Some site shouldn't make any difference.
So we do have, I hope this addressed the question. So we do have a tumor origin algorithm. So if you don't know the diagnosis, you can order that. It's RNA based. It's transcriptome based. So it's not DigPath or H&E. But if we can extract RNA from it, with about 96% accuracy, we can tell the tumor.
Yeah, but we're not even discussing like a CUP kind of thing. I'm just saying, if somebody just sends you a piece of meat, can you tell me what that is?
No.
I guess the question is, why would you want to without the data?
So
I think, well, sorry, go ahead, but Dr. Jane's another question.
Dr. Lockhart was asking that very specific question that I'm asking, which is, let us say I do not have a pathologist on staff and I need a diagnosis. Can I send you a tissue sample? Can you give me a diagnosis of, and with all the details?
So yes, we can do that. Yeah,
Okay.
it's called our TO test, and yes, we can, with about 96% accuracy, we can tell the tumor origin.
Great. Thank you.
Same
Good to know.
diagnosis.
Second question, which is, have you looked at concordance or discordance between the different molecular platforms? Because occasionally I've seen that inadvertently a sample was sent to two different platforms. One turned out to be positive for a mutational profile. Another one did not have the same mutational profile. How much confidence should we have in each of these platforms, and how much concordance or discordance is there?
Yeah, so that answer is gonna probably depend on which company you talk to, but so I've never seen a formal test of two different platforms in a sort of regimented way, comparing the same tissue sample sent to on two different platforms to see what the concordance is. My impression, based primarily on anecdotal data, is that the concordance is very high. It's not going to be 100% for sure, and there are many factors that play into that. The specific slice of the tissue that was sent to the company may vary. Of course, the company's own levels of detection, sensitivity, and specificity for each alteration is going to vary from company to company. But my impression in general is that the concordance is very, very high, but I've never seen a formal study.
Thank you.
Okay.
Thank you so much.
So I was also alluding to this ResNet when I asked about the digital.
I'm right next to you.
I don't think I can speak about it. So when I was asking about the ResNet, this is what I meant. There are like multiple ResNet models, and one is like ResNet 18, 21, 52, 104, and it depends on the layers, how many layers you want to dissect the tissue. And then I'm working on something else in like the malignant hematology world. We're using Res 21 to be able to determine the blast cells. But I was wondering, like, which ResNet are you using, or if there is another model other than ResNet that helps you, formulate the tissue of origin. I think this is the question also Dr. J was asking about.
Yeah.
Just from the pathologic standpoint, without like any molecular.
So I actually don't know, but Joe, do you know what?
I'm writing it down.
Okay, so we'll get you the answer to that. Yeah.
Unfortunately, it's a layer deeper from me.
Yeah.
But we'll get that question addressed. I'm going to put it as an action item. And you said you're working on a project?
Yeah, just like.
And are you looking for like co-development opportunities potentially? Okay, that's good enough. All right, yeah, we'll look into and see and get back to you. Thank you.
All right, kind of out of time. Thank you so much to the Tempus team for
Thanks very much.
talking and bringing lunch. Bringing lunch on board. And again, meeting some of you a little bit later on today. And again, thanks for coming. Thanks for attending. And I don't know if we're having grand rounds next week. We'll see, we'll figure it out. Maybe see you next week.
Thanks. Thank you. So much. Hopefully that was good.
That was great.
Very scarce.
Well, you learn this,
Yeah.
it's like the XTX or that guy, because it's like some of the works that are bringing this.
That was
I had one almost, especially.